Hello everyone. Today we are going to start the next biomolecule, that is nucleic acid. So first of all we should know that what that nucleic acid, where it is found. The nucleic acid is actually was found in the nucleus, okay, of WBC. So that's why, as it was found in the nucleus of WBC, so that's why it was given the name nuclein first, and later on it was understood that it is acidic in nature, so that's why it was given the term nucleic acid. Second thing, what is nucleic acid? Is it a polymer or monomer? Actually, it is a polymer. Okay, so as it is a polymer, so that's why where we will get this polymer in acid soluble pool or acid insoluble pool. So it is found in acid insoluble pool. Next thing, that how many types of nucleic acids are found. Okay, so there are two types of nucleic acids are there. Okay, what are they? They are DNA and RNA. Okay, so where we can get the DNA and where we can get the RNA. See, in case of higher grade of organisms, we have both DNA as well as the RNA. So in case of higher grade of organisms, our genetic material is DNA, but this RNA is performing number of other functions like protein synthesis. But in case of lower grade of organism, the genetic material itself is called as the RNA. They don't have DNA as a genetic material, they have RNA. And RNA is comparatively unstable as compared to DNA. So that's why we can see different variants of archaebacteria or different variants of viruses, as they have the genetic material RNA. They can change their genetic material. Different compounds can be added to the genetic material RNA. So that's why what we can see, that every time they are changing their variants. The thousands of variants we can see, but as the DNA is comparatively more complex in structure, so that's why complex and stable in structure, so that's why it is found as a genetic material in higher grade of organisms. Okay, so remember, it is the genetic material, okay, in higher grade of organism. Okay, and in case of RNA, in higher grade of organism, RNA is found in protein synthesis, but it is a genetic material in lower grade of organism and helps in protein synthesis in higher grade of organism. Okay, now after that, in this nucleic acid, how many type of monomers are there? Okay, so how many monomers are found in the nucleic acid? There are three types of monomers. What are they? They're found, first one, sugar. So there will be a sugar present. Okay, and this sugar is a ribose sugar. So ribose sugar is a aldose or a ketose sugar. So it is actually a aldose sugar. Okay, next one. The second component of the nucleic acid is a nitrogenous base. Okay, so nitrogenous bases are also found in the as the monomer in the nucleic acid, and the third one is the phosphoric acid, and this phosphoric acid is found in the phosphate state in the genetic material. Now, if it comes to the sugars in the DNA and the RNA, we can have the two different type of sugars. Okay, the ribose sugars in DNA is deoxyribose sugar. Okay, in case of RNA, it is the ribose sugar. Okay, second thing, in case of the nitrogenous bases, okay, nitrogenous bases are of two types. One is the purine and second one is called as the pyrimidine. Okay, Purine is double ring compound and pyrimidine is a single ring compound. I will show the structure. But purine, what are they? How many types of purines are there? There are two types of purine. One is adenine and second one is guanine. And the three types of pyrimidines are cytosine, thymine and uracil. So there are three types of pyrimidines and two types of purines are there. In the genetic material DNA, in this nitrogenous bases, what we can get? In this purine, we are going to have adenine and guanine, and from this pyrimidine, we have the cytosine and thymine. But in case of RNA, these purines are found adenine and guanine, but instead of thymine, in this pyrimidine, we have cytosine and uracil. Okay. Next one, phosphoric acid. Phosphate is common in both the DNA as well as the RNA. So these are the two components, DNA and the RNA, where we can get this monomers, that is ribose sugar. In DNA we have deoxyribose sugar. Next one, the nitrogenous bases adenine, guanine, cytosine, thymine and a phosphate group. In this RNA, the sugar is ribose sugar, where we can get this nitrogenous bases adenine, guanine, cytosine and uracil. This is different. Okay. And last one is the phosphate group, which is common to both the DNA and RNA. After that, let's just move to the next part. So in this part, what we will see, whenever it comes to the DNA. Okay, so what is DNA? How does monomers form the DNA structure? See, so whenever, first of all, we'll see the structure, that is DNA. Okay, so DNA is actually made up of two helix, and in case of RNA, we can see only a single helix. RNA can be of three different types. What are they? They are tRNA, then it is mRNA, and then rRNA. Okay, so two types of nucleic acids we can see, DNA and RNA. So these are the two structures. Now after that, we will see that how these monomers, that is nitrogenous bases, then ribose sugar and the phosphoric acid are attached to each other to give this structure. Okay, we will see first of all how these monomers are attached to each other. Okay, so first of all, what is the monomer present? The monomer is the first monomer is the ribose sugar. Okay, so first of all, what is this ribose sugar? See. Okay, so first of all, if it is this, okay, so already I have told you that ribose sugar is a aldose sugar, it's a pentose sugar. It's formed by, it's a homocyclic structure or heterocyclic structure. It is a heterocyclic structure because the ring is formed by carbon, 4 carbon atom and 1 oxygen atom. So that's why it is heterocyclic structure. The fifth carbon is attached to the fourth carbon as a R. Okay, so here we will write CH2OH here. Here it is HOH, here it is HOH, here it is OH, here it is H. Okay, so this is the ribose sugar. So this is the pentose sugar found in the RNA. But if it is the DNA, in the second carbon, the first carbon is this, in the second carbon what happened? One hydrogen, here only one hydrogen is present, not the hydroxyl group. The hydroxyl group is replaced by one single hydrogen. So that's why this will be called as the deoxyribose sugar. So what will be the formula of ribose sugar? In this ribose sugar, it is going to be C5H10O5, right? In this deoxyribose sugar, what will be the chemical formula? That will be C5H10 but O4. So this is a derived monosaccharide. Now if it comes that among these two type of sugars, which is more stable, The stable one is deoxyribose sugar. If one hydroxyl group is present, it is going to be a functional group. It will try to make a bond with the another components. So it is not that stable. But whenever it comes to deoxyribose sugar, it is stable because hydroxyl group is replaced by a single hydrogen. If it comes to that both these two ribose and deoxyribose sugar, which is heavier, okay, or which is going to have more molecular weight, then obviously it will be the ribose sugar, deoxyribose sugar, as one oxygen is not present, so that's why it is going to have less molecular weight, right? So this is the types of ribose sugar. After that, let us see the next type of monomer present in the genetic material DNA or RNA. First one we have seen that are the ribose and deoxyribose sugar. After that, see the nitrogenous bases. What we have seen that there are two type of nitrogenous bases. One is purine, another is pyrimidine. So first one, in the purine we have seen that it is two type. One is adenine, another is guanine, and in the pyrimidines, we have three types of pyrimidines. They are cytosine, thymine, and uracil. In DNA, we have A, G, C, and D as purines and pyrimidines, and in RNA, we have A, G, C, and U. Instead of thymine, uracil is replacing. Okay, now to see the nitrogenous bases, the structures. Sometime maybe in question it may be asked that the structures, you have to just find out which one is that compound. Okay, so first of all, the basic differences between the purines and pyrimidines is the purines are double cyclic compound. It is a dicyclic compound, and in case of pyrimidine, it is the single cyclic or monocyclic compound. Okay, so purines are dicyclic, two rings are syn, and in the pyrimidines, these are monocyclic, having only one ring. Next one, see the structures. One is adenine and another is guanine. This is the adenine, this is guanine, these are the purines. So in this, what we have seen that in adenine, we have NH2 group, amino group is attached, okay, and here in this guanine we are going to see a oxy group is attached. So these are the two basic differences between the adenine and guanine, right? So only this thing you have to remember. In case of this pyrimidines, see there are three types: uracil, cytosine, and thymine. So if you see uracil, cytosine, and thymine, what are the structures here? Once again, here oxy, here we have oxy group, and here we have amino group. If it is uracil, we are going to have one oxy group. In cytosine, we have amino group. In thymine, we have oxy group once again. Right, next one, if you see this different pyrimidines, then in this genetic material DNA we are going to have the cytosine and thymine, but if it is RNA it is going to have uracil and cytosine. The next one is the phosphoric acid. Let us move to the phosphoric acid. After that, just see the next one is phosphoric acid. This is H3PO4. Right, so what happens here with this phosphate, what we can see here, three hydroxyl groups are attached like this. Okay, but this phosphoric acid in the genetic material DNA and RNA, that means in the nucleic acid, it forms the ionic state, or it is found in the ionic state like this. Okay, so this is the phosphoric acid. Now how this whole compound nucleic acids are formed? Okay, so how these nucleic acids are formed, I will show here the all these three types of, that is monomers, that are the ribose sugar, then the next one is the nitrogenous bases and the phosphoric acid, how they are attached to each other. Let's just see. So first of all, the central that is the ribose sugar or deoxyribose sugar, it will be present in this nucleic acid. Okay, so this is the I have not balanced this. Okay, so only just see how the structure will be formed. Okay, balancing part you just need not to know all those balancing part in specifically biology. So what is it? With this first carbon, what we are going to see this dicyclic structure. Okay, so here we have suppose this N. Okay, so this way it will be found. Okay, so this will be the uh, the adenine, and this will be suppose adenine. Okay, so adenine is attached to the first carbon atom like this. Okay, next one, second, third, fourth, fifth. With this fifth carbon atom, we are going to have the phosphate group. So phosphate group is attached like this. So this is the pentose sugar. This is the ribose or deoxyribose sugar. Here we have this one nitrogenous base adenine. So this is a nitrogenous bases. This is one nitrogenous base here, and this one is the phosphate. Okay, so this will be the phosphate. So what we will see that this is going to be the nucleic acid, right? So this is the sorry, here what we will see this genetic material DNA and RNA. What we can see that here this tree will form the nucleotide. Okay, so this will be the unit that will be the nucleotide. Okay, so these nucleotides will be found in complex. They will form the bonds and they will be attached to each other to form a long chain of nucleic acid. So here the nucleotides are the units, right? So this nucleotide is going to have phosphate, ribose and the nitrogenous base. Now in this nucleotide, if you remove this phosphate, okay, and you are getting only the ribose sugar and the nitrogenous base, then it will be called as the nucleoside. Okay, so one thing, just remember the differences between the nucleotide and nucleoside, that in the nucleotide what we are going to have the phosphate, the ribose sugar and the nitrogenous base. But if it is the nucleoside, what we are going to see only the ribose sugar and the nitrogenous base, not the phosphate. After that, see, I have shown here that depending upon the nitrogenous bases, nucleosides and nucleotides are named differently. With the pentose sugar, suppose this is the pentose sugar. With this pentose sugar, if we have the nitrogenous base, that is adenine, then this will be a nucleoside. So this nucleoside will be called as adenosine. And with this fifth carbon atom, if phosphate group is attached, then this will be the nucleotide. So this nucleotide will be called as adenylic acid. Okay. Next one, if the nitrogenous base is guanine, then this nucleoside will be guanosine, and this nucleotide will be the guanylic acid. Next one, if cytosine is present is as a nitrogenous base, then this nucleoside will be called as cytidine, and this whole nucleotide will be called as cytidylic acid. Next one, if it is the thymine as the nitrogenous base, then this nucleoside will be thymidine and the whole nucleotide will be thymidyllic acid. Next one, the last one, if uracil is present as a nitrogenous base, then that nucleoside means with this pentose sugar, if uracil is attached, then it will be called as uridine, and this nucleotide with this nucleoside, if phosphate is attached, then it will be called as uridylic acid. Now just see what is happening with this pentose sugar. This adenine is attached, right? So this bond, okay, so what do you think what this bond is? This bond will be glycosidic bond. Okay, and with this pentose sugar the phosphate is attached, so it will be called as the ester bond. Okay, and this way the nucleic acids are going to be formed, attaching the nucleotide with the next nucleotide. Now after that I will show you how this DNA structure will be formed. So here just see, if these nucleotides are attaching, how they will attach to the next nucleotide, right? So just see, if this is the structure of the DNA, I have told you it is a helical structure. Okay, like this. So how this helix will be formed, right? So here we are going to have 3 prime, 5 prime, and here this will be 5 prime, 3 prime, n. What is that 3 prime and 5 prime indicates, or 3 dash? Actually we should not say it as dash. It is a 3 prime and 5 prime. So just see what is happening here. This is the ribose sugar. Okay, so here I will show, this is the ribose sugar. With this suppose adenine is attached, with this suppose phosphate is attached. With this third carbon, okay, this is first, this is second, this is third, this is fourth, and this is the fifth carbon. So with this there will be another phosphate, and this phosphate is going to make bond with the fifth carbon atom with the next nucleotide. So here we have suppose guanine. Okay, so this way the phosphate will make with two nucleotides, and this will be ester, this will be another ester. So this phosphate will make a phosphodiester bond. Okay, so this way the bond formation occurs. Okay, now with this adenine we are going to have double bond with thymine, with guanine we have double bond with cytosine we have double bond with guanine. Okay, so the structure will be, see, this nitrogenous base is attached to the first carbon atom. So the first carbon atom, if it need to be like this, then this structure will be anti-parallel. Okay, this is free, this is a OH which is found in the third carbon, which is free. With this fifth carbon, phosphate is attached. Same thing is happening for the whole chain. Okay, so here this is a bond formation takes place, right? So okay, so this way what we can see, okay, so I'm just showing here how this bond formation is taking place. So suppose this is a chain. So what will happen to this nucleotide attached to each other to form a nucleic acid? Then what happens here in this first nucleotide, you just see this is the nucleotide having this ribose sugar. With this we have nitrogenous base and the fifth carbon. This is the fifth carbon which is free, right? Now in this chain you just see, in this is the oxygen, this is first, this is second, the third, fourth, and this fifth carbon. So third carbon is free here where hydroxyl group is present, right? So this is the third carbon atom which is free now. Okay, so the next one, if it comes to this chain, then here what is happening, this is the first carbon, this is second, this is third, this is fourth, this is fifth carbon. Okay, so the third carbon is free here, with this hydroxyl group is present having no bond with any another compound, and in this chain towards this end, see, first, second, third is attaching to the phosphate. So here fourth and this fifth, so fifth carbon is now free, right? So whenever you see a nucleic acid, either it is DNA or RNA, then yes, is it showing polarity? Yes. In one end we are going to have 5 prime and another end we are going to have 3 prime end. So this is how actually the bond formation takes place, and it is, see, here we are going to see 3 different types of bonds. With the ribose sugar, the nitrogenous base is attached by, already I have told you, this will be glycosidic bond. Okay, this will be hydrogen bond, nitrogenous base attached to the next nitrogenous base by hydrogen bond, and this phosphate will make the ester bond with 2 nucleoside here. So this phosphate will make phosphodiester bond. So by these 3 different types of bonds, phosphodiester bond, glycosidic bond, and with hydrogen bond the bond formation takes place and the nucleotides will be attached to each other to give rise a polymer, that is the nucleic acid. Either it is DNA or it is RNA. After that, just see the structure of the DNA. In this DNA, this model was first put forwarded by the two scientists Watson and Crick. So this model is also called as Watson and Crick model, as already we have seen in botany. You have already studied this structure, but in very short I will tell you the structure here. This is called as Watson and Crick model, and by X-ray crystallography this model was first discovered, and according to this structure, according to Watson and Crick, this structure is like this. So they had shown us the structure of B type of DNA. So there are different types of DNAs available. Okay, this can be A type, B, C, D and Z. Okay, just nitrogenous base pairs are going to be changing. It can be 10, it can be 11, depending upon that you can see these things, not at all important. So just see, in this Watson and Crick model you can get 3 prime and here it will be 5 prime, this will be 5 prime, this will be 3 prime end. Okay, so here we can see in this DNA structure you can get, this will be a shallow groove, okay, and this will be a deeper groove or deep groove. Okay, in between the two consecutive grooves, it may be shallower to the shallower groove or from deep groove to the deep groove. Okay, from this to this. This will be called as one spiral. Okay, and in one spiral, how many nitrogenous base pairs occurs? Okay, nitrogenous base, here it will be suppose 1. It will make bonding with the next anti-parallel chain. So as it is 3 prime and 5 prime end, it is already anti-parallel. So with the anti-parallel bond, it is going to make hydrogen bond. I already have told you in the earlier structure. So how many? We have suppose 10 base pairs. This is one base, this is another base. So by hydrogen bond, it is make a pair. So base pairs, how many base pairs are there? Suppose it is 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. So here 10 base pairs are present in one turn or one coil or one spiral. What we can call this will be the B type of DNA. So in one turn, okay, or this will be a turn, you can see. So this will be a B type of DNA. So in this B DNA, what we can see, 10 base pairs are present in one spiral. Okay, then you can see if it is this, the length of the spiral is 34 angstrom. And if you see in one spiral 10 nitrogenous base pairs are present, then what will be the distance between two nitrogenous base pairs? Right, suppose from here to here, then obviously this will be 3.4 angstrom. So the space from here to here, this will be called as the diameter. So the diameter of a DNA is around 20 angstrom according to Watson and Crick model. So yes, this is the structure of the DNA. So I hope you understand the structure of DNA. There are different types of DNA you can see, and the types of RNA you don't need to know in this structure, in this biomolecule. But already I have told you there are 3 types of RNA. One is messenger RNA, one is ribosomal RNA, and another is a transfer RNA, which is helping in the protein synthesis. In the molecular basis of inheritance in second year NCERT biology, you will get the structure of the different types of RNA. So in this biomolecule chapter, only up to this much you need to know. So this is all about a nucleic acid. I hope you understand. Thank you.